Reminding me and asking me if I've got the recorder on. I would appreciate that. I just bring them down to Natalie, and Natalie posts them on the website. And so if you miss a class, or if there's something you want to do some reviewing with, those lectures will be posted as an MP3 or MP4, I guess, on the website. Okay? So if we're looking at this scale right here, it shows you the size of different types of organisms and cells. Here's the world we're going to be working in primarily, for the most part. Things that are in the micrometer range. There are some bacteria that are sub-micrometer size, mycoplasms. We'll be talking a little bit about virus, but not much. So what is a micrometer? One micrometer equals what? How much per meter? 1 times 10 to the minus 6 meters. Okay. So small. We've got some pretty good microscopes for you this semester. You probably have used those before in other classes. But the organisms that we're going to be looking with in this micrometer size range are really pushing the resolution of our microscopes. We can get a total magnification maximum with these microscopes of 1000x. And some of your unknowns, you're barely going to be able to see it with a microscope. They're so small. Okay? So one of the things we're going to be working on this semester is developing your microscopy skills and making sure you're very adept at using the microscope to see those little tiny things. So when we talk about the history of the microscope, the history of microbiology, one of the things we'll see is that the early history of microbiology is very closely tied with the development of the history of that instrument, the microscope. You can't really study what you can't detect. So we'll talk a little bit about history of microscopy when we get to that particular point. Well, in terms of types of organisms, depending upon where you take a microbiology course and who's teaching it, what kind of organisms might we be studying that are really small? Bacteria. What else? What is small? Virus. What else you need a microscope to see? You all take zoology, right? Botany?
Some parasites.
There's some parasites. Particularly protozoans. Algae. Not all algae are single cell, but many are. There are some even unicellular fungi. Well, as you can imagine, if we had a course that tried to deal with all these organisms, it would really be impossible. So we have kind of pawned off the algae and the fungi to Dr. Hykins. Dr. O'Neill gets protozoans. We really got kind of a gap. I talk a little bit about virus and cell biology, but we really, if we had room for it, could really use a virology course, because viruses are incredibly interesting and obviously have a lot of medical and also biotechnological applications, but we don't have a virus course. So really, we're going to primarily be concentrating on bacteria. Additionally, this microbiology course, and like a lot of them, also has a pretty strong immunology component. A couple of reasons for that. One is that we don't have a separate immunology course, and yet we need some training in immunology, particularly for students who want to study medical technology. And I don't think, do we have anybody here that's interested? Okay. So you have to have this to apply to St. Francis and IU Health. I don't feel bad about including immunology in a microbiology course because since we're concentrating primarily on bacteria, many of which are pathogenic, one of the things that we'll do is we'll talk first about what adaptations do bacteria have that allow them to be pathogens. And then we'll turn it around and look and say, okay, what kind of defense systems has the human host evolved in order to try to counteract and kill these particular pathogenic microorganisms? So there's a lot of good interplay between these two particular topics. And immunology is really a fascinating topic. All the cell signaling that's involved in immunology, all of the cellular interactions, all of the chemical compounds that your white blood cells produce, including antibodies, which ultimately give you the incredible immunity that you have, and which keeps you healthy for the most part. So, in some respects, if we really had complete truth in advertising, this course really should be called Bacteriology/Immunology. We're exclusively going to be dealing with bacteria in the lab. Okay, we're not going to do any work with virus. We're not going to do any work with any of these other organisms. It's all going to be centered around bacteria and learning the skills you need to work with bacteria, and then learning how to do the various tests that allow you to identify the bacteria. So that whole project will be pretty time consuming in the lab portion. When I was sitting in your position, and for most of my teaching career, we have organized all living things into 5 kingdoms. The person who's giving credit for developing the 5 kingdom system of classification is Whitaker. You know what 5 kingdoms are? I don't even know if anybody's talking about 5 kingdoms anymore, of course. What are 5 kingdoms of life? Which one do we belong to? Animal. Animalia. It's another possible kingdom, you suppose. Plants. Animal, plant. Okay? Any of the other 3 you know? Fungi? 2 more. Prokarya, or the bacteria. And then the protista, which includes the protozoa and sometimes some of these single-celled algae. 4 of those kingdoms have something important in common, and one is different. Which one's different and why? Prokarya, and it's different because those are prokaryotic cells, right? Cell biology flashback. What's the difference between a prokaryotic cell and a eukaryotic cell? No membrane-bound organelles. What's some other differences? Difference in ribosomes, good. 70S versus 80S. Which are bigger? Eukaryotes are typically larger, prokaryotes much smaller. No membrane-bound nucleus. No flagella in eukaryotes, is that true?
I think so.
Ah. Anybody disagree? Sperm cells are flagellated, aren't they? But there is a difference. There is a difference in the organization and the structure of a bacterial flagella and a eukaryotic flagella. Okay. They operate differently. They both have these structures of motility. That's true. Do bacterial cells go through mitosis, meiosis? They don't. Do you know how bacterial cells divide? They divide by mitosis. Binary fission. They literally just split in half. Their chromosome does have to replicate, but then there are two identical chromosomes, and the cytoplasm just divides. Now you've got two cells that are identical to one another. And then those two cells divide, and those two cells divide. And so we can form a population of cells. And what's their relationship to one another genetically? So they are what? Clones. Okay. One of the things that makes bacteria a very, very useful research tool is that they have a very short generation time. Generation time refers to how long it takes a cell to divide and produce a new generation. Instead of hours or days like human cells, it varies, but a typical bacteria generation time is about 20 minutes. And because bacterial populations grow by cells doubling, splitting in half, any population that grows by doubling goes through exponential growth. And you're probably all familiar with the exponential growth curve, which is characterized by slow population growth to begin with, and then at some point you start really picking up speed. That means that on Wednesday in lab, you can take a few cells of your unknown and aseptically transfer them to some fresh, sterile growth media and pop them in the incubator at the optimal temperature for your organism, and come back a day or two later, and it's going to be covered with a confluent film of growth, which will have millions, hundreds of millions of cells in it. Okay? So that rapid generation time is really important, and it's very useful in doing research involving prokaryotic cells. So they're small, no membrane-bound organelles. Evolutionarily, which preceded which? Prokaryotes are the first cells that evolved about 3.5 billion years ago. And we believe that prokaryotic cells were responsible for colonizing early eukaryotic cells by a process called endosymbiosis, and giving rise to two of the organelles that are now associated with some types of eukaryotic cells. What organelles am I talking about? Mitochondria and chloroplasts. Both of which have their own DNA, and both of which have ribosomes. And what kind of ribosomes do they have? They have 70S ribosomes. The cytoplasmic ribosomes are 80S. But the ribosomes of those organelles is 70S, suggesting a bacterial origin, prokaryotic origin. Well, the 5 kingdoms of classification is a concept. It's a theory. It's a model. And hopefully one of the things that you learn in science is that concepts change over time based upon availability of new information. And in textbooks, you don't hear much about the 5 kingdom system of classification anymore. Instead, you hear about the 3 domain system of classification. So here's your old 5 kingdom system of classification. Monarans, protists, animals, fungi, plants. Monarans being down at the bottom, indicating earliest ancestry. And then the line indicates the separation of the prokaryotic organisms from eukaryotic organisms. The three-domain system of classification adds another layer to the hierarchies. And it separates all life into three larger groupings and kingdoms called domains. The domain bacteria, the domain archaea, and the domain eukarya. The domain eukarya is then split up into 5 kingdoms. Whittaker, okay, plants, animals, fungi, protista. So what effectively they have done is they have taken that, what was previously a single kingdom, prokarya, and broken it up into 2 separate domains. There's some real interesting implications for that. We'll talk about kind of a survey of microorganisms a little bit. We'll talk about what kind of organisms are found in the domain bacteria, which ones are found in the domain archaea. What does archaea mean? What's that people say? Ancient. Okay. Archaeology is the study of old things. Okay. So in this domain archaea, there are these organisms like the methanogens, the methane producers. There are these organisms that live in extreme salty environments like the Great Salt Lake or the Dead Sea. Those organisms are called halophils. And there are organisms that can live in extreme heat like the hot springs at Yellowstone Park. Those are called thermophils. So sometimes these members of the domain archaea are referred to as being extremophils. And these are organisms that live in very extreme environments. In the domain bacteria, there are the more typical bacteria that you're used to encountering or maybe have heard of, like E. coli, or staph, or chlamydia, or bacillus. Okay? We're exclusively in lab going to be dealing with the domain bacteria. And all of your unknowns will be in this particular domain. I can't really give you a domain from this archaea because they're really difficult to culture, and we just don't have the tools to grow them and analyze them. Previously, these were in a single kingdom. Now, remember the implications of being in the same kingdom. What does that say? That says a common evolutionary ancestor, and things that are in the same kingdom are more closely evolutionarily related than things that are in another kingdom, right? Okay. Well, look at the implications of now there being 3 domains. Let's take an amoeba and a human. Kingdom Protista, Kingdom Animal. They are in this domain. So what this system of organization tells you is protozoan, or an amoeba, is more closely evolutionarily related to a human than a methane-producing prokaryotic bacteria is to an E. coli. So it's a very, very different way of looking at evolutionary relationships and the origin of life and what kind of cells gave rise to what other kind of cells. Why did we switch from the 5-kingdom approach to the 3-domain approach? Anybody have any ideas? It was new information. What were old classification systems primarily based upon? What determined where you were plant or animal? Yeah, a lot of, kind of visual, gross morphological differences. Okay?
Now we have access to DNA, so if we lose one person.
Bingo, absolutely. Now that we're able to do more genetic analysis, because ultimately, when you look at evolutionary relationships, genes are the greatest reflection of evolutionary similarity or dissimilarity. And the genes that we really honed in on, because they tend to be conserved throughout evolution and don't change much over time, are genes for ribosomal RNA. And so a lot of analysis has been done with ribosomal RNAs and genes. And that information was the basis for the development of this new three-domain system of classification. So when we do our survey of microorganisms, to kind of give you an idea of what's out there, what have we identified so far, why are those organisms important, we're going to be superimposing that discussion on this three-domain system of classification. But at the same time, as we do that, we also want to be able to give you an appreciation for practically how can you go about identifying a microorganism. You're going to get quite familiar with these volumes. These are called Bergey's manuals. And this is the oldest one. This is the 7th edition. This is the 8th edition. This is the 9th edition. This is a set of volumes called Systematic Bergey's. These three are called Determinative Bergey's. And so we're going to have a fairly short but intense discussion of classification from a practical standpoint of identification, i.e. unknown, and then also classification based upon phylogeny or evolutionary relationships. And so it is going to be important for you to understand this new system of classification that has pretty much been universally accepted now. It's in textbooks, and we now talk about the three domains as opposed to the 5 kingdoms. Okay. So that's kind of the what. We could spend a lot of time talking about the why, and I'll talk a little bit about why microorganisms are important today. We'll continue on that tomorrow, and it will be kind of weaved throughout the whole fabric of this course of the importance of microorganisms. Give me some ideas of why microorganisms are important to your life. Okay.
They help with like digestion, stuff like that. They help with body processes.
So there are some beneficial microorganisms, including gut bacteria, which are involved in breaking foods down. Some of those bacteria, as a result of their metabolism, will produce useful substances like vitamins. This is one of the areas where we're really finding out a lot of interesting information about nutrition. For example, ideas about why we have an epidemic of type 2 diabetes. In the past, the conversation has always been about how we metabolize foods that we take in. Should you have a diet high in sugar, or shouldn't you? Should you have a diet high in protein, or shouldn't you? And I mean, you know, diets are all over the place. There's Atkins diets, you know, where you eat only protein. There's things that are high carb diets. I mean, they're just all over the place. But the emphasis has always been about how do our cells metabolize those foods. Now we're starting to find out that what's really probably just as important, if not more important, is how do the gut bacteria metabolize them? And what do they produce as they metabolize those foods? What are some other importances for microorganisms? Right?
A significant amount of pathogens.
Okay, so we've got beneficial activities. We've got detrimental activities, pathogens. We'll spend a lot of time talking about pathogens. But let's stay kind of at the surface for a minute. What are some other importance of microorganisms? Microorganisms have an ecological effect. Some of those effects are beneficial. Some of them are potentially detrimental. Anybody give me an example of roles that microorganisms play in ecosystems? Be more specific. What do you mean?
Organisms.
So decomposition. Is decomposition good or bad? Both? Okay. Can ecosystems be sustainable without decomposers? No, because part of the web of life involves plants extracting nutrients from the soil, and then organisms eat the plants and incorporate that biomass into their tissues. Eventually everything dies. And what would eventually happen to the soil if organisms just died and fell on the ground and didn't decompose? Yeah, all the soil would become depleted of nutrients, all of the key nutrients would be tied up in the biomass of the organisms, and we'd also be piled dead high in bodies of dead things. So decomposition helps to recycle the materials back to the soil again. Can't live without decomposers. Maybe not from necessarily an ecological standpoint, but is decomposition always good? Look at our slides up there. What do we have? We have a bunch of moldy fruit. We have a piece of meat that is covered by this slimy growth, which is probably a combination of a lot of different bacteria, maybe some fungi. We lose an incredible amount of food resources to decomposition, both in the field, in the refrigerator. How many times do you throw out old nasty stuff? There's a lot of economic loss associated with that. And then, consequently, there is a whole huge industry associated with food preservation involving refrigeration, irradiation, and chemicals that we use to try to retard the spoilage of food so that we don't have such great economic loss. Besides decomposition, any other ecological benefits or detriments you can think of microorganisms? Something to tweak your interest for next time. We'll talk a little bit about nitrogen fixation. And how about photosynthesis? Estimated that 50% of the photosynthesis, which produces what? We need oxygen, which then contributes to our ozone layer. 50% of photosynthesis is carried out by single-celled microorganisms. It's not just the trees, folks. Okay, so check your email, look over syllabus, and if you want to print off PowerPoints, you can print them in the lab here. They don't print in color, but it's free. If you want to use this network printer over here and any of the computers, you can. And then lecture notes are available if you would like that. We'll see you tomorrow.
You guys have 3:15, right? Yeah.
19.